Thank you everyone for joining us for Polly's first user group meeting. I wish I could be there to enjoy all the excitement. Unfortunately, I have to be on study section right now while you all are doing this. And I'm going to be telling you a little bit about how we're using Polly to identify new hypotheses and chase after them in ways that we really couldn't think of. And just some background, I'm Richard Kibbe. I'm a scientific co-founder of Elucidata. My background's mostly in metabolism and stable isotope flux work. So I'm coming at you today as a newbie to transcriptional analysis using some of these tools. I'm an associate professor of medicine at Yale, and I'm an endocrinologist where I see patients and am working on trying to understand diabetes and how best to treat it. So my lab's interests really center around intermediary metabolism and its connection to the diseases like diabetes. And some common questions that come up in the lab is, how do beta cells couple glucose metabolism to insulin secretion? Seems like we should know a little bit better by now, but there's still lots of gaps in our understanding. And then how is metabolic homeostasis maintained across multiple tissues? And how do we start to develop the tools to answer some of the finer details of our understanding of this? So, there is a vast majority or a vast availability of multi-omics data, but there's a major problem. There's a challenge that a lot of this data is available, but it's not usable. And we're all very familiar with a lot of the very powerful databases with websites like GTEX and Portal and Geo and the Human Protein Atlas. But in many ways, much of the data is hidden or inaccessible. It's not ready for analysis when you're ready to do it. And it's not easy to consume for a biologist who doesn't have a very strong computational background. It's also cumbersome to store and manage such large data sets, and especially given the variety and the volume of them. And the data lacks structure often, and often doesn't have complete metadata. So these are really significant challenges to try to overcome. And the goal of Polly is to make this data usable. And we basically pump in all the information from them and allow the tools that we've developed online to be able to analyze them and visualize them. So we can take all sorts of different multi-omics data and start processing them to generate the hypotheses that we later go back in and test with more directed methods. So what I'm going to talk about today is a proof of principle pilot use case where we're going to try to understand how we can mine this public data to start to get a better grasp of human physiology. And the database we're going to start with is GTEX, which is the Genome Tissue Expression Project. It's a public resource that was built to study human expression and regulation and its relationship to the genetic variation. And it contains tissue from quote unquote normal donors. And there are a lot of them. There are about 1,000 humans that have contributed tissue to this, and almost 20,000 samples that are present in there. There's transcriptional data among other types of data, and metadata includes sex, age, race, and cause of death. So it's really powerful, but for most of us, we can only approach it through the user interface, which is somewhat restrictive. And so why we brought this onto Polly is to make it more usable. So the data is extant on the Polly library, and that way we can use our own custom designed workflows to answer biological questions that are outside the initial scope of what the portal was actually designed to do. And that's what we're going to talk about today. So I'm going to pose a biologic question that is not easily answerable just using the current GTEX web-based portal. And that question is how can GTEX be mined to understand whole body metabolism? And being an endocrinologist, I like to think about insulin. It's relatively straightforward for me to at least understand. And shown here across the bottom is the expression level of the insulin gene with log fold change across all the different tissues within the GTEX database. And if you look far to the right, you'll notice that pancreas stands out above and beyond all the other tissues. And that's where all of the insulin in the body is made. Now, if you think a little bit more about the pancreas, it's 99% exocrine. Only about 1% of it is pancreatic islets, and then a fraction of that are the beta cells, which actually secrete insulin. So that big signal is coming from a very small fraction of the whole pancreas. And so how can we query this whole tissue to be able to understand something unique about the beta cell and beta cell physiology? So, at a first approach, we can just look at insulin expression across all 328 pancreata that are in the database, and we see that they are highly variable, with the highest towards the left and a gradual reduction in the amount of message as you move towards the right. Now, I've placed bars over these different individuals to suggest what type of physiology may be going on, with actually the normal folks being more in the middle. Those who are insulin resistant tend to augment their islet mass until it starts to fail, and the islet mass or the insulin quantity starts to be reduced as you become type 2 diabetic, or those are destroyed by autoimmune attack in type 1 diabetes. Now, confounders to just looking at the total number of counts of transcripts are numerable. First of all, as mentioned, it's the islet and the beta cells a very small fraction over the overall pancreas. The pancreas auto-digests, and so the sample quality can vary highly from individual to individual. The anatomy of the pancreas is also not uniform. There are more islets in the head of the pancreas than in the tail of the pancreas, and so depending on where you sample from, you may get different responses. And as I mentioned just earlier, there's a lot of variability in gene expression as the disease progresses. And what we're now finding is that some of these samples aren't completely pure. They've been contaminated by other tissues. But still, it is, given the large number of samples that we have to look at, it is really a valuable resource to be able to examine. So what we're looking at in this plot is a correlation of all the other pancreatic genes with insulin content. And you'll notice a surprising thing on this volcano plot, and that is, first of all, that all the genes in the pancreas are positively correlated with insulin. That is, all the significantly changed genes. Now, this is not the typical type of volcano plot that you're used to looking at. So the significance is on the y-axis, and in the x-axis is the variability. And so genes that are highly variable in terms of their expression across all the different individuals are further to the right and all the way towards the left, depending on if it's in the positive direction or in the negative direction. And those with less variability are more towards the center. And here we see that, again, that most of the changes across the whole sample of tissue are largely positive. And while that might be concerning, when you start looking at the information that you're getting at, and especially because you're looking at it across the whole pancreas, it does become more interesting. And what we're seeing is that there are a lot of very expected genes showing up when we correlate it towards the insulin value. I've listed a number of these to the side, and just with this first pass analysis, we're hitting a high number of genes that were picked up in GWAS studies or have been identified as MODY genes, like the sulfonylurea receptor, things that are autoantigens in type 1 diabetes, like glutamic acid decarboxylase, and strong differentiation factors. So the other rather interesting thing is the significance of these hits. When we look at the adjusted p-values of the correlations, they're enormously high. So there does seem to be power in this technique at being able to pick out genes that are beta cell specific, or at least islet related, despite the fact it's buried in the exocrine pancreas. All right, so a key insight from this first mini study is that with a good marker gene, such as insulin, polytissue mining enriches for key beta cell genes, despite being only a small fraction of the overall pancreas. But can we do better than this? Are there different ways of querying this data that can give us even stronger insights? And so this raises a second biologic question that we'd like to get at, and how can GTEX be mined to understand beta cell mass? And so I need to give you a little bit of background to be able to understand this question a little bit. So in the pancreatic islet, there are a number of different types of cells. There are the insulin secreting beta cells, which are indicated in the lavender color here. And then glucagon secreting cells, which are in the blue color. And there are other cell types also present. In a human islet that is healthy, it's generally about 50-50 purple to blue. But as diabetes progresses, you start to lose beta cells, and some of these beta cells actually turn into alpha cells, and you start to have more and more blue cells. And each of these two tissues have independent genes that are good markers for them. So for the beta cell, insulin's a great marker gene, and for the alpha cell, glucagon is. And so what we wanted to do, and what is not typically possible to do, is to create a new gene and put that into the matrix of all these other genes and look at that relationship of our new dummy gene, as we're referring to it, across all the different donor pancreata. And in this case, we're looking at the insulin to glucagon ratio, which would vary as a function of the beta cell to alpha cell mass. And like with just insulin, if we look across these samples, we see that there is a large amount of variability within our donors. But the question we're asking is a little bit more analytically straightforward, because now the normals to the left, the insulin resistant is in the middle, and the type 2 and the type 1 are to the right. So we're looking at the data in a different way, and just to take these same patients and look at just the insulin by itself, just to give you a sense of how they vary and how they may not, you see that insulin by itself is behaving in a different way than the insulin to glucagon ratio dummy gene that we're going to use. And this is seen more clearly on this scatter plot. And what you can see all the way to the left is where insulin and insulin to glucagon ratios are low. These are likely type 1 diabetic patients that we don't have identifier data from in the database. And that is actually what's skewing this regression line. If we didn't have those patients all the way to the left, there wouldn't be a very strong relationship between insulin and glucagon. So now when we look at the correlation of genes across these samples with our new ratio, we see, not unsurprising, that insulin is highly selected for, and glucagon is highly selected against. So our algorithm's doing what it's supposed to do. But now we've shifted a lot of the positive hits to the left, as they actually seem to be localizing with glucagon and perhaps other genes within the islet. And so we're filtering down what we're looking at compared to what we had seen before. And in a quick analysis of these, the 5 top hits in this analysis are all beta cell specific genes. And having gone through a lot of these different portals and looking at correlations of genes within different tissues, this is the first time where I've ever seen anything where we get all 5 hits coming from the same tissue that are specific to that tissue. And they have a significance, even with the Bonferroni corrections. So we think this is a great tool and a great strategy to being able to analyze data by inputting these dummy genes into the mix. And just to compare some of the hits that we're getting relative to insulin alone, there were about 700 quote unquote hits with insulin and 36 with our insulin to glucagon ratio. But a surprising fraction of these were shared. And 11 of the genes that were common to both are listed to the right. And you can see the ones that are in blue are anti-correlated with, and those are alpha cell genes. And the ones in orange are beta cell specific genes, and many of them have been associated with disease states. And some of them have not been very well investigated. So if I were to start looking for a random gene that's associated with islet mass, I think they would start on this list and have a much higher likelihood of being correct than choosing out of 700. Now, if we look for the representation of a number of different genes as it correlates with the insulin to glucagon expression, even though we're looking across the whole pancreas, the power of having this many individuals to refine our data stands out, and it shows that we get type 1 diabetes is being identified as being suppressed in that group. Again, these are supposedly normal people. From the analysis of the data, it did look like there were some type 1 diabetics, and this is the type of thing we can do pretty easily on poly. And we can also look at the pathway maps to get a better sense of what's up and what's down, and does it make logical sense as we develop our scientific questions and our strategies for tackling them. And we can look at things like the insulin secretion map and start to get a better sense of what might be correlating with a high insulin to glucagon ratio or an expanded beta cell mass relative to the alpha cell mass. All right. So then I'd like to take this one step further. And can we correlate genes, gene expression in one tissue, namely the pancreas, with the expression in another tissue? And the question we're trying to pose here is since insulin is a very strong regulator of liver function, and so is glucagon, can we correlate gene expression in the livers of individuals with pancreata in the GTEX database? So what's nice about this database is there are a number of different tissues from different individuals, and there are 328 pancreas samples and 226 liver samples, but 86 of them are from the same donor obtained at the same time. So we now have the possibility to look at the insulin to glucagon ratio in the pancreas and see how things in the liver correspond to that. And our strategy is first to make this dummy gene like we did in the pancreas, filter out the uncommon donors and just look at the common donors of the liver and pancreas and restrict our liver analysis just to those 86. We insert this new gene, our dummy gene, into the liver matrix, and we look for positive and negative correlations with this ratio. And with this, we're able to do cross-tissue transcriptomics. Again, I'm showing you a volcano plot of the different hits that we're getting, and to the left are some of the curated genes I pulled out, which perhaps aren't that surprising. A lot of lipid-related genes and other metabolic genes in the liver are correlating with a high degree of significance with the insulin to glucagon ratio in the pancreas. And this is with a relatively small number of individuals. And so we can start to do this with different genes and different tissues and even across the whole database. And that is really, the power of being able to use Poly to develop your own workflows to mine some of this data. So in conclusion, rather than letting the platform drive your questions, Poly enables the question to actually drive the platform. And what I've shown you here is a proof of concept work in progress where we looked at single cell-like analysis from bulk tissue. We were able to pull out insulin-related genes from the whole pancreas. And then set up dummy gene queries, which can be all sorts of different type of analytical approaches for narrowing down or challenging the data to pull out unique insights. And we're also able within this database to cross into subsections of the database, look at one tissue relative to another for intra-individual cross-tissue analysis. And so Poly has made this usable with its ready-to-use data libraries, custom-designed workflows, to query biological questions outside the initial scope of the portal. And perhaps most importantly, it's biologist-friendly. It's something that I can do with just a little bit of assistance. And with that, I'd like to thank the assisters who really made this possible. And this includes Sandhya and Shafali, who were an important part of this presentation. And I won't be able to personally take any questions, but I'm sure our folks at Elucidata will be able to take some for you. With that, I thank you for your attention, and I hope this was helpful for you.